Okay, so I'd like to welcome everybody to our 14th, but NHABON community practice webinar. And I'm noticing that my camera doesn't appear to be working, but I'm obviously here if you can hear me. The need for offshore observations to support HAB warning, a case study of the West Coast of the US. And so you can see down there for technical issues, contact Masha Edmondson. And this webinar is being recorded and will be available at the IUS Association website under the NHABON tab. And I think that Masha will put the website in the chat or the Q&A, the chat, I think. So let's see, can we have the next slide? So I am the Quay Dortch, the co-chair of the NHABON Community of Practice and also the Senior HAB Scientist at NOAA. And my fellow co-chair is Dr. Clarissa Anderson, but since she is going to be a speaker today, I am mostly going to be doing the introductory material for this webinar. So first, I'm going to do a little bit of an introduction, and then there are going to be two presentations, one by Clarissa and one by Vera Trainer. Then we'll have a Q&A discussion of the webinar, and then we will wrap it up with a discussion about a schedule for future webinars and meetings, etc. So, could I have the next slide? You all have seen this before, but I wanted to remind you that the mission of a National HAB Observing System is to provide for HAB detection, early warning, and forecasting. And for that, you need offshore observations as well as nearshore and shore-based observations. Could I have the next slide? Yeah. So I put in here a very short list of some of the most advanced observing systems that we have at present. So one of their main purposes is to provide early warning and, if possible, prediction. And offshore observations are needed for a variety of reasons which vary between regions. Bloom initiation may occur offshore so that shore-based sampling does not catch the early stages. Blooms may stay there or come inshore, and the observations are needed for early warning. Offshore data are also needed for model validation. So as you can see from the descriptions of these systems here, I've put in the kinds of sampling that are currently being done. And coastal cruises are listed for all of these, but they provide very limited data. For the obvious reason that most of you are familiar with is the difficulty of getting the offshore data either by coastal cruises or in some cases satellite remote sensing. Ships are hard to schedule and they're very expensive. It's particularly hard in an event response mode for using ships. Remote sensing does not work for all species and in all regions at all times. Thus, there is very poor offshore coverage in most cases, and shore-based sampling could have better temporal and spatial covering as well. So could I have slide number 5? As a result of this, all of these observing systems and many others are instituting or experimenting with new technologies to improve spatial and temporal sampling.
Masha, can you advance one slide? That was just an animation.
Yeah. So there are many new sensors and new platforms, and it's basically been an explosion of technologies. These pictures are just a few of the new sensors and platforms that I picked out of the earlier NHABON webinars. Thank you to the earlier presenters for the use of their figures. The new technologies focus mostly on rapid automated detection and quantification of cells and toxins, often remotely. New platforms avoid or minimize the use of ships. Moorings allow greater temporal coverage. Surface autonomous vehicles and underwater autonomous vehicles make greater spatial coverage possible, sometimes tracking a bloom, sampling thin layers, or responding to an event. Some of these technologies are in the really early stages of development, but show great promise. As all become more routine, the capabilities will become even greater. It is an exciting time to be developing observing systems. However, there are many hurdles to fully implementing the offshore sampling that is needed for NHABON. A major one is that most of these systems are dependent on research dollars. There is funding for a few NHABON pilot projects, and that is a start. But keep in mind that we do not yet have an operational National HAB observing network. For this webinar, we will be focusing on the West Coast today for two main reasons. One is that the bloom hotspots are well known, which makes offshore sampling easier. Not that it is easy. And two, new technologies are being actively implemented in this area. Can I have slide number 7? So the two speakers today are Clarissa Anderson and Vera Trainer. Vera Trainer is the Aquatic Sciences Director of the Olympic Region Harmful Algal Bloom, or HAB program, at the University of Washington's Olympic Natural Resources Center, as well as an affiliate associate professor at the University of Washington. Her current research on harmful algal blooms focuses on the assessment of climatic factors that influence toxic bloom development and intensity and impact the resilience of coastal economies and marine wildlife health. Dr. Clarissa Anderson is a biological oceanography with expertise in ecological forecasting and remote sensing. After a 2-decade tour of just about all the University of California campuses and a short stint in DC, she is now the executive director of the Southern California Coastal Ocean Observing System, or SCOOS, at Scripps Institution of Oceanography, where she continues to conduct research on phytoplankton ecology and HABs in coastal California. She is an elected member of the UNESCO SCORE Global HAB Scientific Steering Committee, the UN SCORE Working Group on Observing Air-Sea Interaction Strategy, the Science Advisory Team for the California Ocean Protection Council, OPC, the U.S. National HAB Committee, and serves as the co-chair of National HAB Observing Network Community of Practice. So Vera Trainer is going to start out. We request that you put your questions in the Q&A, and they will either be answered by the speakers if it's a short answer, or they'll answer it after the talk to the rest of the audience. So Vera, if you want to go ahead and start.
Yeah, thank you so much for that nice introduction, Quay, and good afternoon, everyone. I'll be telling the story about the Pacific Northwest and specifically how we are using offshore monitoring to help supplement our early warning of harmful algal blooms in the area. This is thanks to many, many people, but I just want to acknowledge first our funding sources, NANUS, NOAA IUS, ORHAB, and also NOAA and COS. Okay, let me see if I can advance here. Okay, but first a little bit of background. I'm sure many of the folks in the audience know this already, but just to make sure we're starting from the same baseline, there have been a lot of news articles about domoic acid on the US West Coast, including this one from 2015, where it was documented in the Seattle Times that this might be the largest bloom ever. The toxin itself, domoic acid, is produced by certain species of the genus Pseudonychia. Some of them are very, very intense domoic acid producers, and some produce only a little bit. But when that cell is fed upon by marine organisms, for example, Dungeness crab seen there on the left, or planktivorous fish such as sardines and anchovies, they can make their way up the food chain to poison people. But in this case on the right, you see a sea lion that's being intubated because it ate too many sardines and anchovies laden with domoic acid. So there can be very serious effects of this toxin. This map has been seen in many, many places. This is the result of NOAA funding, where we were able to go on a multitude of cruises in 1998 that really gave us a nice snapshot of what can be seen on the US West Coast. These are discrete samples taken by Niskin bottles at all of these stations. So everywhere you see a dot is a station. And the more intense colors are where we saw the most domoic acid. And what you'll notice is that there are these hotspot sites, the Juan de Fuca Eddy, Hecata Bank, and then moving further south. So I'll be focusing more on the hotspot sites there in the Pacific Northwest, and Clarissa will focus more on the sites in the south. And you can see some recent news headlines that document some of the impacts of these domoic acid events on this coast. But first, I wanted to mention, in discussing with our Indigenous collaborators, that when I showed them these maps, they say, oh, we know about this. This is the prairie. The Juan de Fuca Eddy is known by the Macaw tribe to be a very rich source of marine life. So it's an area where they are able to do whaling at times. In the present day, they historically did whaling, and it's known to be a very productive site. But among the plankton that reside in this area are some of these toxic phytoplankton, including Pseudonychia. So the basis of the partnership in the Pacific Northwest, and here I'm focusing on Washington, is the Olympic Region Harmful Algal Bloom Partnership and the Oregon Partnership. And I'm happy to say that Oregon just received funding for an expert to do the sampling in the area. You may have heard that over 40 people got sick due to paralytic shellfish poisoning in the state. So it takes illnesses in order for the funding to be found. But at any rate, the ORHAB program is shown here. And you note the sampling sites in the brown circles, and that highlights the collaboration of a number of tribes, the Macaw, the Hoh, the Quinault, and the Quileute. And then also Fish and Wildlife does a really important job of sampling the southern coast. And then the Olympic Region, the Olympic Natural Resource Center, a part of the University of Washington, also assists with management of that partnership. Where you see in the red, these are the razor clam beaches, which are an important subsistence harvest for the tribes, but also a really fun recreational harvest for the general public. So this forms the basis of the sampling that's done in Washington and then also in Oregon. But what we know through modeling, and this was work done by Hallie Stone, that toxic cells from these hotspots, and the maps on the right show particles released from the Juan de Fuca Eddy hotspot site in 2017, 18, and 19. It takes up to 10 days for these cells, these particles, to actually reach the beaches. The trajectories shown in green are the samples that actually reached the beaches. So we can provide an even earlier warning to managers by knowing what cells are available at these hotspot sites. So how are we doing this? This is one of the tools that we were just able to acquire through the IUS Ocean Technology Transfer Partnership. You see our lovely lightfish here on the left in the yellow. The usual light fishes that are acquired by the Navy are dark gray, but they're excited to be working with oceanographers that want their light fishes to be seen. You see Anthony Odell opening the hatch, and that is where the water sampler is placed. The samples collected by the lightfish need to be analyzed in a lab very quickly in order to be used in the Pacific Northwest HAB Bulletin, and I'll be mentioning more about that in a little bit. So this is a lightfish mission. They are from a day to 2 days. The timing is coordinated with the bulletin dates. The sampling locations are generally based on chlorophyll A satellite images. We target 15 samples per mission, and we also have a variety of environmental parameters, sensors that are on the lightfish itself. Here's an example of a mission out of Newport, Oregon, that was guided by satellite imagery. You see in the center there, the imagery near Newport off the Oregon coast, showing Hecate Bank, and then the imagery in Washington on the right. But not every bloom hits the coast. So how are we doing that sampling? Here you'll see some drifter tracks showing in blue the drifter tracks during upwelling periods, and in red, the drifter tracks during downwelling or storm periods. So we need a combination of fair weather to allow the blooms to propagate, but then also storms, reversal of winds to allow the cells to then make their way to the coast. So how do we know that this is happening? We're using, for example, the environmental sample processor, which is shown here. And let's see, you're all in my way here. In the pink star there, you'll see the location of the ESP, the current location of the ESP, and it was strategically placed in the track of the drifters from the eddy center. So it is our hope that blooms originating from the eddy will make their way past the ESP, which is able to then provide real-time or near real-time toxin and cell concentrations through a variety of manipulations around this carousel that you see in the center image. So the ESP you see on the left there, it has cell and satellite modems that transmit the data as images to a shore-based server, which is then interpreted by researchers at NOAA and then placed on the NANU site for incorporation into the Pacific Northwest HAB Bulletin. Another tool used, and as Quay mentioned, our cruises are few and far between, but when we have cruises, this is a really useful tool for rapid high-resolution imagery and to be able to know whether Pseudonycha are present in the waters that are being sampled, as you see on the right in the images here. So there are a number of different data pools that are put together to create explicit forecasts, which then put together are synthesized by Ryan McCabe and others to create a bulletin HAB risk. And I'm going to go into a little bit more of that and show you what the bulletin looks like in the next couple slides. But you see that offshore data are an important component of this work. So here's the first page of the bulletin, and this was requested by managers of the Department of Fish and Wildlife and Department of Health. They requested that we leave all of this information in there, but they said they're very busy people. They wanted our risk assessment front and center at the top of the bulletin so that they can either read the bulletin at their leisure or they can act quickly if the HAB risk is red, for example. So in this particular example, you see the risk rating is green, and we've used data from phytoplankton samples and the seawater toxins, both from the beach sampling, and I'm showing here in red the offshore samples, which will increase with time now that we have the lightfish and other tools employed. On the right are the data from the ORHAB partnership and also our Oregon partners. We also include climate, winds, river flow, because the Columbia River can act either as a conduit or a barrier of these harmful algal blooms, the weather, currents, and satellite chlorophyll, and then the ocean forecast, in this case, the live ocean model. This is then all put together in a summary text that's put together by Ryan, Barb Hickey, and myself. So what do these offshore samples give us? They give more time for managers to act. And with the blue dots here, or sorry, green dots, I show examples of management actions. Collection of additional samples, which is typically the first step. Continued harvest with increased confidence. And there have been cases of extended harvests, for example, in spring of 2018, increased harvest limits in 2017. We went from 15 to 25 clams a day, resulting in many happy clammers. And then there's been cases of harvest postponement, delay, or closure. But it's been very rare in recent years that all beaches are closed, that we have coast-wide closures, largely because our forecasts are so good. And you can see quotes from coastal managers, and I'll just read the one from Joe Schumacher from the Quinault tribe. Access to weekly HAB monitoring and guidance from the HAB bulletin gave confidence to go forward with the tribal dig. So you can see the HAB bulletins at the NANUS website, nanus.org, and the ORHAB website, which is now orhab.uw.edu. I encourage you to take a look at all of the past bulletins. This work couldn't be done without a huge number of people that put together the teams. I'm showing here the Lightfish team, the ESP, and the IFCB teams. So thanks to many, many people. Here is the basis of the monitoring. This is a photo from one of our ORHAB annual meetings. So thanks to these folks who really initiated a lot of the work that's happening in Washington State and now Oregon. And thanks to our funding sources, IUS, NCOS, and then in-kind funding from all of the coastal tribes who are able to fund their staff. And this is the reason why we do what we do. Thank you.
Yeah, I love that. What area do you live in?
Shall I just launch in?
Nice job, Vera. Yeah, I'll pass the torch on to you, Clarissa.
All right, thank you. Let me know if things don't look correct. It looks okay on my end. So I will follow up on Vera's really nice overview of her region with a little bit of a deep dive into what is happening in California. Many of you are aware, but I think it's really good to highlight the need for offshore measurements and the value that that brings both to our early warning systems and then also to the science that we do and our understanding of how these blooms initiate and then even terminate. So let me just click through here. Some of you have seen this slide before. It's worth noting that what we're calling now maybe like a holistic early warning system for HABs in California really has its origin in the early 2000s as we started to really understand the gravity of Pseudonychia blooms in particular and domoic acid events and their impacts on wildlife, ecosystems, public health. So as that system evolved, first in academia and then kind of extended into the more boundary organization space with groups like SCOOS and SENCOOS, it has really evolved to include many different components of shore stations, routine sampling, there's state sampling, there's models, many different types of models, and we even have a bulletin. So I can't touch on all these things today, but we're trying to look at this as a prototype system and understand the gaps and understand where we're failing when it comes to particularly early warning for these HABs and their impacts. So to remind you, you've seen a bit of impacts for the Pacific Northwest. With California, the impacts are a little bit different north to south. There's definitely more of an emphasis on fisheries like Dungeness crab in the north. And then when you get into Southern California, huge rookeries for California sea lions. Not to say they're not throughout all of California, but we really start to see some hot spots for those rookeries as we get further south. And in general in California, adult female strandings for sea lions are a sentinel for domoic acid events. But Throughout the state, bivalves are impacted by these toxins. Pseudonychia is by no means the only one, and we do heavily monitor for saxitoxin and other toxins. But sort of getting into that area, those hotspots, Vera showed you that nice map from the Hake cruise, and it may have popped out that Santa Barbara Channel is a known hotspot, particularly from cruises like that, and even work done earlier, that there seems to be something about the Santa Barbara Channel that leads to the initiation of a lot of these blooms early on in the season, particularly because when upwelling winds are blowing from the west, which happens earlier in the season, you get upwelling in the Santa Barbara Channel because of the curvature of the coastline there. And so I'm showing you a map here. This is a really old paper, but it illustrated that there are these cyclonic eddies within the Santa Barbara Channel that are seasonal. They're fairly offshore, and they play a role in concentrating and even promoting productivity of diatoms like Pseudonychia, and then enhancing that toxicity within those eddies. So this was some of the early work where we thought, okay, there is an offshore component to this, and how do we incorporate that into our monitoring? Flash forward many years later, we worked with, in more of a modeling context, we were working with a group of stakeholders along the entire West Coast to understand the needs that are out there across a whole sector of issues, harmful algal blooms being one of them. And this is sort of before we were thinking about NHABON, but it fits nicely into the ways we're trying to have a kind of a co-design and user requirement approach. So two of those things was, of course, lead times. We need better lead times. 2 weeks would be beautiful. Predicting offshore bloom initiation became one of the real big topics that was brought up. And I thought that was interesting because I think as scientists, we understood it was a problem, but it became clear from many of the end users we were speaking to that they understood that this was a gap in our knowledge, and at least a gap in our sampling. And so if you look back at sort of the different solutions we've had to early warning systems and trying to get spatial and temporal coverage at fixed sites, in California, the HAB map group, which really became codified in 2008, and Skoos and Senkus took it over as a sustained project, but working with our academic partners throughout the state to keep this sampling going. It's weekly, and it has really great resolution in the sense that we're looking at a whole suite of taxa and toxins, mostly just domoic acid, but we have information on other toxins and certainly on nutrients. And I think it's been really useful, but it does miss things. And we're starting to see that more and more as we get into a pretty high domoic acid event episode here in California, where we've had many of these in a row. And it wasn't really a surprise to see some of this variation in cross-shore variability, because we have seen it before. Different fits and spurts of sampling projects in the Santa Barbara Channel, mostly on things like Plumes and Blooms cruises, have shown us that from the shore station perspective of HAB map, like you can see here, Goleta Pier or Stearns Wharf, relative to samples offshore, seven stations here as part of Plumes and Blooms, even when you have something near shore, does not mean you don't have something offshore, and vice versa. And we've started to see that kind of mismatch quite often, and even in the vertical variability, where you would have a lot of domoic acid or Pseudonychia or both in the subsurface, but not at the surface where a lot of the sampling was being conducted. And this jives with a lot of other studies out there. Dave Caron's work on upwelling of Pseudonychia populations from those thin layers and chlorophyll maxima. We see this in the Bight in particular, but you also see it in Monterey Bay and other places. And so in the most recent spate of events that we've had in 2022, when this really got going, and each time this has happened in the last 4 years in Southern California, we've seen a lot of this stuff start in the Santa Barbara Channel. In this particular case, the time series here in the left plots are showing you that we really never exceeded bloom thresholds for Pseudonychia species, or really saw any domoic acid at those shore stations throughout the whole duration of almost, I would say, about a 1.5 month long domoic acid event. But we were able to get on some cruises of opportunity. In this case, this is a Cal Coffee cruise that was going through the Santa Barbara Channel and hitting the center station, that would be the Plumes and Bloom Station 4. And what we saw was the highest domoic acid offshore and subsurface from that sampling. And this is actually a Plumes and Bloom sample, but we have Pseudonychia image from Cal Coffee. So we're kind of doing a whole bunch of these different things simultaneously. And lucky that we have that, because we wouldn't have known that these sea lions were really acquiring the toxin mostly in the subsurface. And this is out of reach of most of our routine sampling. So now this last year, in 2022, 2023, 24, and then this year, 2025, we've had these major events. But if you're just looking at the time series from the shore stations, like that you see in the colors here in the map on the upper right, you're not going to capture a lot of that variability, and you're going to miss a lot, and you're not going to always have the early warning that you want, because even if we do capture peaks in things like Pseudonychia seriata class, the toxigenic group, or domoic acid, we're not often capturing them before we start to see sea lions come in. They ultimately are the sentinels in a lot of these events, because the blooms are starting offshore. They're acquiring toxin offshore, and we're really only seeing some of the nearshore manifestation later. That's not always the case. Sometimes we see a little bit of an uptick nearshore. That happened in 2025. I don't know that we were monitoring as closely as we could have. It really wasn't until sea lions came in that we realized there was an issue. But I want to just point out that we don't have a lot of great synoptic coverage, but what we do have is a model that was instated as an operational model in 2018, CHARM, and it gives us something. It's like Hui said, satellites aren't perfect. We can't always see everything from satellite. Our detection methods are not perfect. This is very statistical, but it has been able to give us some kind of a synoptic picture of what it looks like in the California current during one of these events, and helps us relay that information to marine mammal stranding groups in real time. And it seems to match up well with that kind of integrated signal that the marine mammal populations, particularly California sea lions, particularly adult female California sea lions, are integrating over that spatial domain. But a lot of these things all have their limitations, so I'm trying to point that out in each of these slides. You know, we still need a lot of in situ validation, and we're not getting that. And it could be in the form of moorings, other Lagrangian platforms like gliders. You saw some cool things from Vera. There's also Sea Trek, like Mike Brosnahan is using. So many options out there. And in the case of this most recent bloom, which really kind of got going in Baja in December, and then has really expanded into about a 2-3 month bloom throughout California, getting almost as high as Bodega Bay at certain points, but really focused in Southern California. We used that CHARM approach quite a lot to communicate and understand ourselves what the evolution of the bloom looked like. And eventually did see a lot in the nearshore zone, and it was very important, but it wasn't necessarily the thing that helped us convey how things were changing in the broadest area of habitat for the animals as this evolved, since impact to sea life has been so massive in these last few events. And we're trying to understand that too. Maybe another webinar for another day. I'm gonna get rid of this, not now. So in this, if you look at this through the lens of CHARM across these 4 years and the number of animals impacted, there is a little bit of a mismatch there. You don't necessarily see that these averages are fully aligned with the mortality magnitude of each of those events, but it is still the sort of main synoptic picture that we have for offshore. Another solution, though, is sort of to bring that information into a bulletin context, a lot like what Vera described. We're doing this for all of the different data streams and the CHARM model, and using it as another way of communicating to end users how to understand cross-shore and along-shore variability, temporal and spatial, in these various metrics that we use for understanding HABs. And this is not a forecast bulletin. The model is sort of the forecast. In this case, this is a retrospective. There we go. Next slide. So thinking about this through the lens of NHABON, where we've had pilot projects going for a number of years now, California has been one of those pilot projects. And so working together, SCOOS and SENCOOS, with all of our academic partners and many pioneers in the field here, we've been able to implement this imaging flow cytobot network with the hopes of not just getting real-time information at those same shore stations where HABMAP takes place, but also getting out on the shelf and getting some information that might really help us understand what's happening in the offshore environment and in places where we know are hotspots for HABs. Oh my goodness, my computers. Getting new computers is never fun. So what we've seen with this imaging flow cytobot, it's in theory 9 operational instruments, up to about 12 in total on Cruises of Opportunity and other platforms, is that when you have them all running and you can look at a nearshore site and an offshore site simultaneously, you're going to see pretty broad differences. I point out Santa Cruz Municipal Wharf and the power buoy that's offshore in Monterey Bay. one time slice, just to give you an idea of how different those populations can be, where you can see a fair amount of Pseudonychia offshore and maybe only a Cashewo inshore. And so if your only metric were a nearshore alert, you would miss some significant events, most likely. And we've seen that when we apply classifiers to the image data. Clear here that with these weekly average data, if you're looking at another site where we have an offshore mooring on at Del Mar, which is about 3 miles offshore, and then Scripps Pier, which is our nearshore site in San Diego, that even during the known Pseudonychia event that we had, as it was transitioning to something more like a dinoflagellate-dominated population at the shore station, Pseudonychia persisted offshore. And while this might not point to the origin of the bloom, it certainly points to variation in how these blooms terminate, where you might have either bloom sources or refugia. And so lastly, I would just say that there are many other ways to handle this. I really enjoyed Quay's slide with the photos of platforms that you can use. A number of eco-HABs have been going on in California to apply these different techniques. One is a modeling technique where you just say, can we get our best estimates of something like domoic acid through a mechanistic model and use that as a way to have the kind of spatial coverage that we need. Another way is to send out these long-range AUVs and sample blooms in situ as they are forming, and you can do that with a number of molecular techniques now. And some of the early evidence that we've seen there that this is important is that we've seen blooms really starting in fronts offshore in Monterey Bay. Again, just pointing to the need to have this kind of information. Will we ever have a fleet of these things out there sampling all the time to tell us when these things evolve? No, but I think that the more we have them at the ready and funding that can get things out there where we're not scrambling to do it every time there's an event would be really helpful. It would cost some money. It will take resources. And so I'll just end by saying that there are a lot of ways we could do this. We have many platforms and cruises of opportunity we're already leveraging, but those cruises and platforms are themselves highly leveraged research projects. And we don't have a lot of dedicated funding to understanding these offshore blooms, their initiation, keeping the funding going for the research to understand how to predict them. And oftentimes we go to NCOS for event response money when these things happen. And I think it's been an incredible resource, but I would like to look at ways to harden these systems so that we can do a lot of this before the bloom starts, or be ready to tackle it very quickly once one does begin, and encourage a lot of interoperable data that allows us to understand the gaps and ultimately fill the gaps. So I will end there. Thank you very much. Okay.
So thank you for some very interesting talks. Mark Suttleson is going to take over for me as the moderator. You all know him as the NOAA MARHAB Program Manager. Yeah,
Yeah,
I can
great.
see you now. Good.
Yeah, great. Thank you. Okay, so we've got a few questions that have come in so far. Thanks for those. The first one, Clarissa, is to you from Marco Sandoval Bomar. Do you think that in recent years, the events have tended to occur more offshore compared to past decades? And are we at a point where we can begin to relate this shift to broader climate modes like ENSO or PDO?
Yeah, thanks, Marco. Glad you're here. I don't know for sure if this is more offshore in recent years. I don't know that we have enough data to say that. I wish I could answer that with some certainty. It does seem like a lot of these blooms have generally had an offshore context. I mean, we certainly did see that in the first decade where we were sampling this a lot in the early 2000s. And in the particular case of 2025, this event has been associated with a major La Nina. We've seen other La Nina events in the past associated with these. A a lot's been said about other aspects of ENSO. I think there have been some tight correlations with El Nino years. So statistically, I think it's challenging to say if it's any one of those particular. I do think this decadal variability matters, or other kinds of low frequency variability in the California current matters. And I think it's connected to the nutrient variability that's being upwelled in the California undercurrent. So we need to look at those things. We also need to look at food webs. I mean, I think I'm interested in how the bottom up and the top down are coming together. And if there's variability driving the bottom up from like a low frequency variability perspective, it's probably also doing something to the food web, and the two together may lead to understandings of impacts. And so I think you're spot on. We've got to look at that, and we are looking at it, and you're one of the people looking at it. So thank you.
Yeah, thanks for that answer, Clarissa. But I wanted to also see if Vera has any similar thoughts on the same question. And essentially, it's, Vera, do we have any indications from work that you've been involved with as well about climate, ENSO and PDO and their relationship to domoic acid or?
Yeah, I mean, we've looked at some of the data, especially the data collected in Oregon, and we've seen a strong correlation during positive PDO years and domoic acid events, warmer, anomalously warm years and PDO and domoic acid events. So yes, we believe that they're going to increase with climate warming.
Great. Thanks both. I appreciate that. So, another question's come in from Mingxun Zhang. I'm not familiar with California HABs or California HABs, but why does it seem a high probability of toxin in nearshore if the blooms are generally initiated offshore? Is this because when blooms reach offshore, it's generally under downwelling conditions or low nutrients?
Okay, I'll try to unpack that. I'm not
Yeah.
100% sure I understand the question, but yeah, if we were to sort of think theoretically about an upwelling cell where upwelled water is coming up onto the shelf to the near shore and then being pushed offshore and invected offshore and eventually maybe pushed down, you have maybe like an Ekman transport figure in your mind that might explain some of this. But it does seem as though a lot of the vertical transport of upwelled water on the shelf is often happening very intensely offshore. We're talking within, 20 kilometers. I'm not talking way offshore in the California current. But our sampling site is at piers. And so we're going to miss that until conditions are such at those pier sites that we also start to see the plankton responding to those upwelled waters. So it's not as though the conditions are totally different in these different places. It's just a timing issue. Others may have a different perspective on this, but that would be my answer for now. Thanks.
Thanks, Clarissa. I appreciate that. We have a following question, or another question's come in. Kyla Kelly asks, it seems like Washington has a good understanding of how toxic cells reach the coast from offshore hotspots. In California, do we have a similar understanding of how blooms are reaching the nearshore environment after starting offshore?
Yeah, we don't have one particular, thank you, Kylie. We don't have one particular place where, like say a Juan de Fuca eddy, that's sort of a known hotspot, origin spot for these blooms. They're kind of often starting all over the offshore areas of the coastline. And it's not exactly like those particles then get invected into the shore. It's more like the conditions evolve such that in places where we have pier sampling, we start to see the plankton responding in kind to those same conditions as the conditions spread. So I don't see it as a completely similar situation. We've done modeling studies and advection studies where we're not necessarily seeing particles moving around and then hitting a beach. It's more as though conditions seem to come up offshore that are conducive to bloom and domoic acid development or in the subsurface and eventually evolve to spread throughout this cross-shore zone. I'd be happy for anyone else in California to say something. Maybe Rafe has a thought. That's how I see it based on my gestalt understanding of California.
Thanks, Clarissa, for that. I appreciate that.
Rafe, do you have anything you want to add to that since you've been looking at it longer than I have?
I think you can unmute yourself, Rafe, if you're
Yeah, I
available?
am.
Yeah, there you go. Thanks.
Yeah, so I think Clarissa covered it well. I think the big difference is the Pacific Northwest has sort of a unidirectional flow. And so blooms kind of build up in the Juan de Fuca eddy often and then get burped out and moved down the coast. And if the Columbia River isn't in the way, they hit the coast. And we don't have that same scenario in California. It's a much more detailed coastline. We don't simply have flow from north to south or even onshore to offshore.
Great. Thanks, Rafe.
Mark, I just want to mention, it says there's one question answered, and I actually failed to answer it online. But maybe
Which,
you want to read it from, well, I can read it from Esther. She says, what are the physical conditions characterizing the nearshore hotspots? Are there any common features among them? And I can take a stab at that.
sure. And we'll get back to the other ones. So that are
Okay,
before
and
that,
they're
sure.
they're very physically retentive sites, Esther. So for example, the Juan de Fuca Canyon or the Juan de Fuca Eddy, they're underwater canyons. There's flow from the California current system and also out from the Straits of Juan de Fuca that make it a counterclockwise gyre. And I think similarly, Hecate Bank in Oregon, it's a very retentive site where there's upwelling going up onto the shelf.
Thanks, Vera. We're going to come back to Morgane's question. Morgane McKibben has asked or made a comment. Thanks for the great presentations, both talks. Note the utility of satellite data in contributing big picture information to offshore HAB monitoring. Morgane's curious on the speaker's thoughts as to whether new ocean color data products, such as those from NASA's PACE mission, may potentially better supplement offshore HAB monitoring compared to current data products.
I'm happy to tackle that.
Thanks, Clarissa.
Thanks, Morgane. Yeah, I think, because the spatial resolution won't be too different from the past satellites we've used, temporal roughly the same. I don't think we're going to make huge strides there, but because we have hyperspectral with PACE, I think those of us working in that area of this HAB space have high hopes that there will be enough research devoted to this area that we can have better detection algorithms that get us towards higher fidelity of whether we're looking at a specific organism or a specific toxin than we've been able to do optically prior to now. I think that by and large, though, having satellites like PACE and it's the legacy satellites that predate it have given us the kind of synoptic coverage we need. Clouds aside, we'll still have those issues. So I think it's a great question, and it's a little bit TBD. We're all trying to understand if it will allow for drastic improvements in our detection abilities.
Thanks for that, Clarissa. And maybe that's something that we can take back into our steering committee discussions for maybe a future webinar for NHABON. We've got a few folks who are part of our team who could help address that. And Morgane, thanks for that question. Maggie Broadwater, colleague here in NCOS, is compliments you guys both on great talks and appreciates the nods to EcoHab contributions. Clarissa, can you discuss the frequency, intensity, duration of inshore impacts beyond marine mammals, such as shellfish, crab closures, concurrent with observing toxigenic pseudonychia blooms offshore without concurrent inshore observations? I think you touched on that briefly, but yeah.
Thanks, Maggie. Why the zingers? Why? This is hard. Okay. Let me try to tackle this without being too reductive, and I'm sure I will be. I think what's tricky, and I'm going to try to base this off what I think when I'm observing the data that I look at, that it feels like, especially in many of the recent events, we would see the animal impacts before we would have any kind of shellfish impacts. In 2022, we never even really had those shellfish impacts because I don't think we ever saw any aspects of the bloom near shore. But there have been other years where we saw the animals come in, we then sort of saw some uptick of this stuff at near shore, and then the shellfish advisories kind of trickled out after that. So it does speak to maybe some kind of an offshore to nearshore evolution in conditions and blooms. And it's not to say that these crab or other shellfish closures aren't happening simultaneously, but they do seem to lag those events. But that's not always been the case. We've had years where we've only had some impacts that were manifested as shellfish advisories and didn't see a lot of marine mammals or birds coming in. And so this, again, speaks to variability in whether these blooms start offshore, start near shore, start offshore, move inshore, only happen inshore, et cetera. Does that answer your question?
Okay, great. If it doesn't, it's odd, Clarissa. They can't say yes or
If it
no.
doesn't, she's coming for me.
So it was an excellent answer to the question. But if you have follow-ups, please either post in the Q&A or come to us directly afterwards. Thanks. Vera, I believe you addressed Esther's question. Marco has a follow-up, perhaps a basic question, but since Clarissa briefly touched on it, he's curious, how do we define offshore in the context of these events? Is there a particular distance, threshold? Is it defined more dynamically based on oceanographic features? That's a great question.
It's yet to be determined,
Yeah.
Marco. Maybe this is your next paper. I think you need to look at that over the sort of suite of data that we have, because oceanographers, other types of scientists who work in the marine environment use these terms very differently. And when you're more on the modeling side, like you are, you end up looking at some rather large domains, and we sort of lose sight of the fact that even 3 miles off the coast where the Delmar mooring sits, I'm calling an offshore site, which would look near shore in your resolution, the resolution of the models you look at. So I think there are things happening on the shelf. There are things happening even outside the shelf. And the few times we've had the opportunity to get those kinds of data, like on a Cal Coffee cruise where we did sample hundreds and hundreds of kilometers offshore, we did see some domoic acids. So there's stuff happening there. And I don't think we properly understand exactly how far out these populations go before you get into more oligotrophic waters. How often are things initiating that far off? Is 50 kilometers a better understanding of this based on nearshore upwelling swaths, or should we be going out to 100 kilometers? I don't really know the answer.
All right, thanks. There was a question earlier that disappeared, but I found it from Terry King, just asking if groups had looked at data with the lens of the 18.6 year lunar node. And then Terry also followed up with a publication, a follow-up paper, in case people don't know what that cycle is in the Northeast Pacific. I will maybe somebody can grab that and share that with the folks in a way that gives some context to why that paper is being shared. That'd be awesome. Maggie, just to follow up and thank you. And Rosa Hunter made a comment that they're in Whatcom County, that's in Washington State. She's noted there are a few beaches that are closed currently due to PSP, paralytic shellfish poisoning. I come to the Julie Matwiu has posted a question from Kodiak, Alaska. Thanks for the presentations. Vera, can you speak to Pseudonychia dynamics in Alaska and whether they've seen significant cell toxicity in Alaska waters? Julie knows that nearshore shellfish have not tested above the regulatory level and that we've seen low DA levels in marine mammals. How much do we know about these blooms, particularly offshore in Alaska?
Yeah, Julie, you guys need to invite me back to Alaska. But I will mention that some of the Alaska folks have recently reached out to Jerry Borchert at the health department here in Washington, and they are starting to see domoic acid in shellfish. I think more work needs to be done. My understanding is most of the monitoring that's done, or a lot of the monitoring that's done, is close to the commercial areas, although I think it's increasing in more recent years. A variety of species, and clearly there are highly toxic species in the area. So it looks like it's an emerging problem there in Alaska.
Yeah, thanks, Vera. I'd echo some of these discussions and points that Clarissa has made, you have made about what's happening offshore with Pseudonychia blooms and toxicity may never actually be observed at the coast. there's this time lag potentially, or there could be different pathways. We're still learning all of that. So any data that we can get as we're learning more about, or seeing some toxicity in various regions from offshore, I think could really go a long way to help us better understand what's happening. Ondine Morgan Knapp has posted this question. Again, thanks. You guys are getting lots of kudos for your great talks. Cool there. Question about the spatial patterns of toxicity in the Pacific Northwest, given the activity of the Juan de Fuca hotspot and eddy. Does it seem to be the case that the more northern stations pick up on toxicity earlier than southern stations? And if so, on what order is the time lag? Days or weeks? Do you want to grab
Yeah.
that one, Vera?
Yep, sure. Good question, Undine, and I'll answer it with it depends. But what we found is that in the springtime, when we have more storms and the flow is more toward the north, we're seeing that cells reaching the Washington coast are generally, and Oregon coast are generally coming from Hecata Bank from down south in Oregon. And then when we reach the fall season, we're seeing cells coming from the Juan de Fuca eddy source. Of course, this is an overgeneralization, but as you saw from the drifter maps, it can take up to 10 days from these source regions for cells to reach the coast. Of course, it depends then also on what the Columbia River plume is doing. And as Rafe mentioned, it can act as a barrier to cells actually reaching the coast. So it's a complex situation.
Thanks for the answer, Vera. Julie confirmed it and agreed with comments that there's still more to learn about offshore blooms in Alaska. I think our questions have petered out, which is great. We might give another second for the hamster wheels that are the internet to post any questions might be lagging. Okay, we've got a few more minutes scheduled for the webinar, but while we're waiting for questions, there's a slide that, I don't know, Clarissa, do you want to handle this one?
Yeah,
Just to
happy.
share some announcements for the community. And if you have another question, put them in there. Thanks.
Thanks, Mark and Quay. So the next NHABON webinar, topic TBD. We welcome thoughts and ideas for topics, rough dates sometime there in September. We want to announce maybe a little early, but next US HAB Symposium is in October 2026 in Cedar Rapids, Iowa. You can go to this link and learn more about it. And if you're interested in sponsoring, please let us know. We are definitely looking for sponsorships. The next international meeting, ITCHA, is in October in Chile. Register if you haven't yet. But I wanted to call attention to this workshop that we have where we're going to describe our draft implementation plan. Oh my goodness, here's my phone. The draft implementation plan for the next 10 years of Global HAB and the Global HAB Scientific Steering Committee. So we really want your input as the HAB community on that. Please come if you are at the meeting. Thank you.
Okay, great. Thanks for those announcements, Clarissa. All right, I've got two more questions so far. Esther Garcia has asked, are the offshore and nearshore populations of Pseudonychia in California genetically identical?
So a lot of work is being done on that. I would say no. Some of the beautiful omics works that's coming out of Cal Coffee Cruises from Andy Allen's team, and Stephanie Wood is leading a lot of this, is really interesting. I mean, the number of taxa and some identified, many identified taxa that are out there is pretty shocking. And there's quite a lot of variability south to north as well as cross shore. And so we're by no means capturing all of this with our light microscopy and our IFCBs. We're seeing these large morphotypes, but there's a lot of other stuff out there. Whether it's toxin producing is a whole nother question. Likely not, but there are big differences. And Holly Bowers is also looking at this, and I think it's kind of a burgeoning area of research. So thanks for the question.
Thanks, Clarissa. Question from Bruce Wright up in Alaska, working with the Knik tribe. They've collected nearly 1,000 samples this year, mostly for PSP or paralytic shellfish poison testing. Can you give advice on the best time frame for 2025 sampling should be analyzed for DA? Samples should be analyzed for DA. They seem to have DA events later in the year in Alaska. Vera or Clarissa?
Yeah, I can give that a go. Hi, Bruce. What I would suggest is that if you have samples collected for microscopy at the same time, to choose those samples where you actually have pseudonychia. And, we have typical threshold levels of, I think it's 1 million cells per liter of the large cells and 20,000 cells per liter of 1,000 cells per liter of the small pseudonychia and 20,000 of the large. That's when we start analyzing for domoic acid. And I'd be happy to chat with you more about that. Send me an email.
Thanks, Vera. All right, we have, I think, come to the end of the questions. Yeah, again, thank you both, Clarissa and Vera, for excellent talks, and thanks to the folks in the audience as well for sending in excellent questions with 1 or 2 zingers. And I think we've got some more information on this slide here about the NHABON program that was discussed and some additional contact information. We do have a bit more time scheduled for the webinar, but I think we may be coming to the end of our questions and what do you think, Clarissa, Vera, Quay? Should we call it?
Yeah, I think people are starting to leave, so I would say
Yes.
Yeah.
time to wrap up.
Yep.
Great. Well, thanks again, everybody. Great job, speakers.
Yes, thank
See
you
you
very
next time.
much.
Thank
Thank you. Take care.